So we're talking today about organelles, and the next set are it's the endoplasmic reticulum. Such a long word, endoplasmic reticulum. We usually just call it ER because it takes too long to say otherwise. There are two types, and you can see them here in this picture. This one looks smooth, and we actually call it just that, the smooth ER, the smooth endoplasmic reticulum. This one has ribosomes on it, and they're bumpy, so we call this the rough endoplasmic reticulum. Do you remember what ribosomes do? They make proteins. So the endoplasmic reticulum is a network of hollow membrane tubules. They connect the nuclear envelope to the cell membrane. So they span the whole cell, pretty much. They function in making cell products and in transport. So they'll modify the cell products. The smooth stuff is mainly involved in lipids, and the rough stuff with all these ribosomes that make proteins is involved in moving or modifying proteins. So this is just an enlargement of it, showing the two kinds. The smooth one over here and the rough one is this stuff. And again, it's just if you look at it under a microscope, the smooth one looks smooth, the rough one looks rough. So the rough endoplasmic reticulum, we'll take that first. The rough ER. That's the one with all the dots. Looks rougher. If you look at it with a real picture, a real electron micrograph, here's the smooth stuff, and see the rough stuff has these dots. It kind of looks rougher. Those dots are ribosomes, and the ribosomes are the things that make proteins. So it would make sense that the rough ER has something to do with transporting and modifying proteins. So proteins are made by ribosomes on the ER surface here. There are actually ribosomes that are free in the cytoplasm, and they'll make proteins too. But the proteins made here usually wind their way inside and then are transported around through here. And they're often modified, meaning they'll take a few amino acids off, or they'll add a lipid or a carbohydrate or something onto those proteins. The proteins are then threaded into the interior of the rough ER. So the protein's made here, and then it moves into here, and then it's moved wherever it needs to go. So the proteins are then threaded into the interior of the rough ER to be modified. So add a little bit of maybe a few glucose molecules onto it, or add a lipid onto it or something, and then move it, transport it to where you need it to go. Smooth ER, that's this part, that's the stuff with no ribosomes. If there's no ribosomes, then it doesn't have anything to do with proteins usually. So smooth ER lacks ribosomes on its surface, and it looks smooth, so that's why we call it smooth ER. The functions, it makes membrane lipids usually, and other lipids. Let me go backwards a little bit. This smooth ER is also involved often in detoxification, so you have a lot of it in your liver. Okay, ribosomes. Ribosomes are I think this is the first non-membrane bound organelle that we're talking about. Ribosomes are made out of two things, proteins and a special kind of RNA called ribosomal RNA. Oops, it says it right here, doesn't it? So they are made of proteins and of ribosomal RNA. That R stands for ribosomal, because they make ribosomes. They are not membrane bound, just protein and RNA, all mixed up together. They function to join amino acids together to make proteins. So this thing right here is actually messenger RNA, so we'll call that mRNA. This big thing is the ribosome. This thing right here is actually another type of RNA called transfer RNA. And these things are amino acids, lysine, glutamate, whatever, amino acids that are being joined together. So the ribosome's job is to take amino acids, put them together, and you know that the building blocks of proteins are amino acids. If you put amino acid together to amino acid, to another amino acid, to another amino acid, you get a protein. And so DNA codes for RNA, and this is the RNA. And the RNA codes for a protein. And so here's that protein being made. These three little nucleotides will code for one particular amino acid. These will code for another and another. So that determines the amino acid sequence. It's so important. We spend a whole unit on it. You get to learn all the enzymes that do it and a whole ton of stuff about that. So this whole process where you're making proteins is called protein synthesis. So you need to definitely learn the word. If you haven't by now, the word synthesis means to make. So this whole process is called making proteins, protein synthesis. Ribosomes can be free, they can be attached to the rough ER, so you see the ribosomes attached to the endoplasmic reticulum here, or they can actually just be free. So they can be free or unattached floating around in the cytoplasm. So if they're attached to the ER, that protein probably needs to be modified and will be sent somewhere. If they're free, those are just the proteins that are floating around in the cell that do stuff for the cell. Free ribosomes make proteins that will usually stay in the cell, whereas membrane-bound ribosomes, ribosomes attached to the ER, will make proteins that will be incorporated into the cell membrane or sometimes sent out of the cell. Sometimes you need proteins out of the cell, like collagen, for example, to make your bones strong. So I like this picture because it shows the ribosome. There are actually two subunits of ribosome. This ribosome is physically made out of proteins and ribosomal RNA. Here's the messenger RNA, and these three will code for one amino acid, and these three will code for another amino acid, and these three code for another amino acid. And look, here are the amino acids. The amino acids are already, look at this. These amino acids are already starting to loop. That's the secondary structure, already starting right away. How cool is that? So anyway, we'll do a whole unit on that, so don't stress about it too much yet. Oh, movie time. The movies never work. Okay, Golgi body. Also, these are called the Golgi apparatus. So here's a real picture of it. This is just a fantastic dead-on picture of it. I like that one. And this is a diagram of one. What you might notice are all these little vesicles. So the vesicles join and the vesicles leave once the materials go through the Golgi body. So the Golgi body's job is to sometimes concentrate stuff. So these vesicles might all come from the endoplasmic reticulum with a lot of stuff in them. All that stuff will be concentrated or modified through here, maybe some protein added to some lipid or whatever. By the time they come out here, they're ready. They're ready to either be sent out of the cell or sent to whatever job they're supposed to do inside the cell. So the Golgi body looks like stacks of flattened sacs. That's what they are. They receive proteins made by the endoplasmic reticulum, and then they transport vesicles with modified proteins. Oh, sorry, transport vesicles with modified proteins, pinch off the ends. So this one just pinched off here. This one just pinched off from over here. Transport vesicle. So the job of the Golgi body or the Golgi apparatus is to modify, meaning add a carbohydrate to it or something, to sort and to package molecules, usually from the ER, into transport vesicles and to transport them out of the cell or to use somewhere else in the cell, so somewhere within the cell. So this is really, you can call it well, in my day, you'd call it shipping and receiving. Let's see, EI, right? Receiving, is that right? The shipping and receiving part of a factory. Of course, there's very few factories in our country anymore. You might want to call it the UPS of, I don't know, of whatever it is. So you're getting stuff in and you're sending stuff out. And this thing will decide where it's going to go. And it's a very cool animation that you probably can't see. So I'll show you that in class. Okay, lysosomes, I'm going to continue because lysosomes actually are one of the things that come from the Golgi body. So here's a Golgi body, and there are some vesicles that have come off of it. So the vesicles came from the ER, and then they go through here, they get concentrated or whatever. Some of the vesicles will end up leaving the cell. Some of the vesicles, well, and spitting their contents out of the cell. Some of the vesicles are lysosomes, and they have a job inside the cell. The lysosome's job is to destroy stuff. It's like the recycling center. So they contain digestive enzymes, and they break down stuff. So they'll break down food, for example. So if you're a bacterial cell, the lysosome will break down food. Digest ingested bacteria, so you can use those as food, or if you eat plenty of good food. The bacteria is something you need to get rid of, so the lysosomes will get rid of bacteria for you. They'll get rid of any cell that has a virus that has infected it, and hopefully they'll get rid of toxins, and they even get rid of cells that have cancer sometimes. So they break down non-useful tissue to get rid of waste also. So lysosomes, think of these as the recycling center. Recycle. They get rid of all the junk that you don't need, whether it's non-useful tissues, just old tissue that you don't need anymore, or if it's bad stuff, bacteria, viruses, cancer, toxins, it'll get rid of all that stuff for you. Another really great video that I'll show you later. So the idea for lysosomes is that they're there for you to break down stuff. So who would have lots of lysosomes? And the answer there, which cells in your body? The answer there is that your white blood cells would have a lot of lysosomes in them because the white blood cells eat bacteria and viruses. The lysosomes join with those. So I'll show you that in a little bit. Endomembrane system. This isn't one organelle. It's just the idea that all these organelles work together, at least the Golgi and the ER and the lysosomes and stuff. So a system of organelles that function to produce, store, export biological molecules. So they degrade junk, harmful substances. It includes the nuclear envelope, the smooth and the rough ER, lysosomes, vacuoles, transport vesicles, Golgi body, and the plasma membrane. So everything except, maybe not the ribosomes because they're not membrane bound, because we're talking about endo meaning within, and membrane, and ribosomes aren't a membrane. But pretty much everything else, except maybe the mitochondria and the chloroplasts, because they have their own special jobs. So the endomembrane system, so for example, if you have a protein made on a ribosome, it might go through the ER and be brought to the Golgi. Maybe this will be a lysosome. Maybe this one will be brought in a transport vesicle, and look at this, expelled out of the cell. Because sometimes you need something like an antibody sent out of the cell. Here's another vesicle that's just going to store stuff. Here's a lysosome. Sometimes something is sent over to the smooth ER to get some lipids put onto it. And I think, let's do one more. Let's do peroxisomes. So peroxisomes are membranous sacs containing enzymes. They're like lysosomes, but they're there really to digest stuff. So they might have catalase in them to break down hydrogen peroxide. So they'll break down fatty acids and amino acids, for example. And they detoxify the bad stuff that's in your body, anything toxic. Okay, and I think that's where we'll stop for now.